Welcome to variables and chi-square tests. We're going to talk first about some different types of variables. So in terms of variables, we're usually as scientists interested in two variables, and we're often interested in how one variable might influence another. In that case, one variable is called a dependent variable, and it depends on the other variable. These kinds of variables, dependent variables, are usually placed on the y-axis, the up and down axis. The other kind of variable is called an independent variable, and it doesn't depend on the other variable. Usually that variable is placed on the x-axis. This will all make a little bit more sense in a little bit. So in terms of thinking about the different kinds of variables, there are continuous variables, which are measured, and they can have any range between 0 and infinity. So for example, this little chick is 3.1 inches tall and weighs 34.16 grams. Those are both continuous variables versus discrete variables, which are another kind of quantitative variable. It can be a number, but it's some kind of a count. It's an integer. So this little octopus or squid has 8 legs and 4 spots. Those are discrete variables. Both continuous and discrete variables are quantitative. They are measurable or countable, okay? That's in contrast to what are called categorical variables, which are words, right? So I'm a turtle, I'm a snail, I'm a butterfly. Okay, those are variables there, and they don't have any order to them. They're just words or categories, right? Versus ordinal categorical variables could be like small, medium, large. There's an order to them, or I'm happy, I'm okay, or I'm unhappy, right? And binary variables are two things that are mutually exclusive. I'm alive or dead. I'm, here or not there, right? So those would be binary variables. So to summarize, the different types of variables can be quantitative and continuous, things like temperature or moisture, things that you would measure along a smooth interval. Things that are discrete, anything that you can measure using integers, numbers of individuals, how old you are in years, right? Those would be discrete variables. Or they can be categorical variables. And these are variables that are not numbers, things like colors or successes and failures, yeses and nos, presence and absence. And categorical variables can be nominal, just names with no order. They can have an order to them and be ordinal, or they can be binary. Now, binary variables can also be continuous, especially if you've studied computer science. One of the most famous binary variables is 0 or 1, right? Okay, so that's kind of setting the stage for variables. Now, we're going to dig in today on categorical variables mainly. So categorical variables, think in your head about some examples that you might be interested in. Well, how about things like color or species identity or habitat type? So is it, an ocean, a stream, or a pond? Mortality, yes or no? Presence, absence of different species. So these are some examples of things that we can measure using categorical, basically using categories. So consider, for example, a warming experiment. Looks like my animation didn't start up, so this is a little bit busy slide. So imagine a warming experiment where you either go out and you warm or control. So you have these plots, and you either apply warming or they're a control plot, which means there's no warming. And in each plot, there's a bunch of lupins, and you randomly select 20 lupins. And in the warmed plots and the control plots, you count whether they have white flowers or purple flowers. In this case, which variable is independent? Which variable is dependent? What types of variables are these? And if there was no effect of warming on flower color, what would you expect for the results? Now, I'd like you to pause the video and try to answer these questions. And I'll give you the answers next. Pause. Okay. So, which variable is independent? Well, warming versus control is independent. That's something that you're setting up with your study, and you get to determine it. Now, you're thinking that maybe flower color will depend on whether the plots are warmed or not. And so in that case, flower color would be the dependent variable. Both of these variables are categorical, warm or control, white or purple. And if there's no effect of warming on flower color, what would you expect? Well, if warming doesn't influence flowers, then you'd expect the same numbers of white and purple flowers in both warmed plots and control plots, right? Okay. And then remember, you put the dependent variable on the y-axis and the independent variable on the x-axis. So if we plotted warm versus control on x on the horizontal and color, white or purple, on y, then what we would see for what's basically called, if there's no effect of warming on flower color, this is called our null hypothesis. What do we expect if there's no influence of one variable on the other? That's called a null, N-U-L-L hypothesis. So here's basically a picture of what the null hypothesis is proposing. If there's no effect of warming or control, then we'd expect about half white and half purple flowers in both treatments, okay? Just randomly, there's no effect of warming, and so the flower colors, the expectation is that they're going to be kind of equal and the same in both types, okay? So this would be the null hypothesis. And you can see the warm and control is on the x-axis, and you can see white versus purple is on y. Okay, now, what if when we warm the plots, we get an increase in one kind of flower color or another? Okay, so this might be the actual results, right? So what is this showing us? This is kind of a weird plot, right? But if you just break it down, and this works with all graphs, and you look at what's on the x-axis, and you can say, okay, this is my independent variable, and it looks like it's influencing flower color in this way. The warmed plots look like they might have more purple flowers, right? And the control plots look like they might have more white flowers. So this is showing that possibly the warming treatment has an influence on flower color. Now we'd want to run statistics to go along with this to see if it's actually a real pattern or if it's just something that might have happened due to chance alone. And that's what you're going to learn how to do today using what's called a chi-square test. So chi-square tests are used, they're very useful when your dependent variable is categorical, okay? You use them when you have some observed values, and you either know what the expected value should be, or you can calculate an expected value from your data set. We'll go into the details of that. You can also use them when you want to test some kind of an association. So it's really fun in plant communities. Down in the Sonoran Desert, a lot of saguaro cacti seem to grow with a nurse plant association, like a shrubby plant, like a Palo Verde. And so you could do a test where you determine, do saguaros tend to grow more where there's already a shrub present or not? So it's looking at kind of dependence between two variables, or an association between two plants in this case. So at the core of chi-square tests, you're trying to compare some observed value to some expected value. Okay, so let's use some other examples. Is my milk in the fridge really 2%? Like, my expectation is that it's 2% milk, but if I measure the milk fat, would I find that it's 2%? Okay, so that's an example of an observed versus an expected value. This is something that I do a lot in my research. I wonder, do leaf litter mixtures decompose at rates expected based on each species in isolation? Or do they decompose at some faster rate when they're mixed together, or some slower rate than we would expect when they're mixed together? You're going to learn some in genetics. You're going to learn about Hardy-Weinberg equilibrium. You can use a chi-square test to see if populations are in Hardy-Weinberg equilibrium. And you're going to learn, or you might learn about Mendelian genetics and the ratios that you would expect if a trait is breeding true, or if there's really, if the expectation that we would see for heterozygosity and homozygosity due to Mendelian genetics. And you can also see if a particular organism, you can test for whether their habitat preferences would be expected based on the amount of habitat that's available, or if they're showing some overwhelming preference for a particular habitat, even when it's not very available. So we'll go through some of these examples today. Okay, so here's just the 2% milk example. I feel like this one's pretty easy to understand. Okay, so you go out into your community and you say, in Olympia, is our milk 2% milk, or is it different from 2%? Okay, so you collect lots of different samples of different brands of milk. Okay, you measure the actual milk fat content in the lab, and then maybe you get results like this, right? Different types of milk, A, B, C, D, and E, and these measured milk fat contents. Well, 1.98, 2.07, some of them, 1.67, they seem kind of variable, right? Now, what do you think you could do to test whether these milks are different from 2.0? Any ideas? I mean, are you worried that that none of them are actually 2.0? Are they all wrong? Like, is this not true? Is 2% milk a lie, right? Well, probably not. There's got to be some variation, right? So, but is the variation around the 2% that we're seeing in these samples, is it okay and they're all roughly 2%, or is it like really extreme and we should be worried? Okay, that's kind of what we use a chi-square test to help us understand. We can't look at the data and know, right? That's what statistics is for. We can't see the patterns and the noise of the data, but the statistics help us to see the pattern. So what we might do is compare each of those values to 2.0 and see how far off they are. And then maybe add those values all up, right? And see overall how deviant is our milk versus a 2% expectation. That's exactly what a chi-square test does in general. Okay, so we're going to walk through this example so that you understand it. I'm going to use some calculations that might at first seem scary, but hold on, okay? So first of all, I'm going to talk to you about summation notation. And this is just a little bit of a review, hopefully, or maybe you've never seen this before, and that's okay. So summation is shorthand for just add it all up, okay? So for example, the Greek letter sigma, see this kind of this big shape here, sigma, that's a capital letter, sigma, and it stands for add it all up. So if I said j, some variable j equals the sum of 1 plus 2 plus 3 plus 4, what would j equal? I'll pause the video, add it all up. Okay. J equals 10, all right? If I then said k equals 5, L equals 20, and M equals the sum of J plus K plus L, pause the video and calculate what is M. All right, so M should be 35, right? So, and then, oops, there's a little infinity symbol there. I'm going to get to it in a minute. In this milk example, 2% milk example, let's say each milk sample is an observation, O, all right? And our data set is made up of a set of observations called O sub I, with the little I denoting which sample I'm talking about. And I can have between 1 and infinity samples in my data set, okay? If this is the case, then we could write our milk results this way. O sub 1, O sub 2, O sub 3, O sub 4, and O sub 5. Each of those O's is an observation of the milk fat content, okay? And the little I is a number between 1 and 5, all right? So we could compare each of these O's to an expected value, E sub I, which would always be 2.0, okay? So keep in mind, we've got O sub I's and we've got E sub I's, right? Those are gonna come into play into this calculation that's coming up next. Here it is. It looks a little scary, but let's break it down. Okay, so first of all, you see the big summation. And what it's telling you is we're going to sum from I equals 1 to n, where n is our sample size. And in this case, n is 5. We have 5 samples. So we're just, all this is saying is add things up from 1 to 5 through your data set. O sub I is equal to the observed values of milk fat in the table, right? So if we're going to add up every value, O sub I minus E sub I. E sub I is 2.0, right? we're going to take those values. It's kind of the deviant, like how devious is our milk fat. O sub I minus E sub I, we're going to square it, and then we're going to divide by E sub I. Now we have to do this for each value of O, right? And there's 5 of them. So this gets a little bit messy, but keep in mind, we have the sample size, O sub I, E sub I, and chi squared is the Greek letter X is chi, not chi, but chi. So the chi squared equation is this. And so let's look at the milk fat example and we'll walk through slowly and it should make sense. So you can see the equation up there. Just keep in mind, I know it's big and ugly, and there's our data. O sub I equals 1 through 5. Okay, so chi square equals the sum from I equals 1 to 5, and then we'll take the first value. The first value, O sub I, is 1.98 minus 2 squared divided by 2, right? Then we're going to add the next one, 2.07 minus 2 squared divided by 2. And then we're going to keep doing that, okay? Plus 2.19 minus 2 squared divided by 2 plus 1.67 minus 2 squared divided by 2 plus 2.22 minus 2 squared divided by 2. And if we do all the math now and we add all these things up like the summation is asking us to, then we get a value of chi squared equals 0.0994. I want you to hold on to that number in your head or write it down, and we're gonna learn about what that means, that chi-square value, after a break. Okay, so please take a 10-minute break, and when we come back, we're gonna learn more about chi-square tests.